All right, so the last thing we got to do is discuss the physical properties and the reactions of amides. We already talked about one of the reactions that form amides, right? So those condensation reactions. Okay, so just keep that in mind. That is a reaction that we've already discussed. So first of all, amides can have that same primary, secondary, tertiary designation. Okay, and how do those come into play? Well, specifically in terms of melting points and boiling points, our primary amides have the most because they can form, this NH2 group can form the most hydrogen bonds. Secondary amines are slightly lower because they form fewer hydrogen bonds. And tertiary amides, they can't form hydrogen bonds with one another, so they have the lowest melting point or boiling point. Notice that here for a while we've been talking about boiling points, and all of a sudden now we're talking about melting points. They're one and the same. So if your trends in boiling point are this compound's the highest and this compound's the lowest, it's going to be the same thing for melting points as well. The reason that the book switched to melting points here is because amides tend to be solids at room temperature. And so typically we characterize them by melting point more than boiling point like we would a liquid. But again, the trends are the same. So whether I asked you for melting or boiling point, you would still go back to your same logic as to what's higher than what. Okay, so we can see, so this is a list of melting points. You can see that the primary amides, so let's actually draw all of these out so we can make sure we understand primary versus secondary versus tertiary. Propanamide, so that would be 3 carbons. That would be a primary amide. N-methylethanamide, so this now has 2 carbons. This would be a secondary amine. And N,N-dimethyl-methanamide, this would be 1 carbon attached to the nitrogen that's then attached to 2 other carbons. And this would be a tertiary amide. So again, you're talking about how many carbons are the nitrogen bound to. If it's just the carbonyl carbon, it's 1. If you have another carbon on the other side of the nitrogen, it would be secondary. And lastly, if you have 2 other ones, it would be tertiary. And again, we can see the trends in the melting point that are given. The primary amine has the highest, followed by the secondary, followed by the tertiary. Again, we see the same trends with boiling point, but because these are amides, the boiling points are much higher, okay? In fact, of all the functional groups that we've talked about, amides have the highest boiling point, primary and secondary amines, even more than carboxylic acids, okay? So in terms of ranking our different functional groups, primary and secondary amines have the highest boiling points of all the functional groups that we're going to study in this semester. Okay, so keep that in mind. Comes to solubility in water. We have similar sort of considerations with the NH2 group from a primary amine can form really nice, strong hydrogen bonds with water. So if you have fewer than 5 carbons, we're always in the same sort of range, 5 or 6 carbons. For amides, it's 5 carbon cutoff. We say anything less than that is going to be soluble in water. Okay. Secondary amines, they can form fewer hydrogen bonds because they just have that 1 hydrogen. They can still form hydrogen bonds with water though, so we're again kind of got that similar cutoff of 5 carbons or fewer. It will be soluble in water. When it comes to tertiary amines, they can only have this, they can only accept a hydrogen bond with water. And so first of all, they have the lowest melting point as we saw, because again, they just don't have that donor hydrogen. But yeah, they'll only form 1 hydrogen bond with water. So they are not as soluble as primary or secondary amines. Okay, the simplest amide in that occurs sort of in nature, and actually the very famous molecule in the history of organic chemistry is what's called urea, which is an amide that's excreted in urine. It's famous because it was the first molecule that was both discovered in the human body and made in a lab, right? So for the longest time, they thought that human bodies were special and they didn't share the same chemistry as every other compound. So when they did this, they sort of proved that actually everything going on in here is just like what they've been studying outside of the human body. So notice it's an amide that has now 2 nitrogens on either side. It's its own special little compound. Again, we talked about how amides can be found in medicine. And so aspirin and a lot of aspirin substitutes rather, contain amide groups. So acetaminophen and Tylenol is one of them. I don't know what phenacetin is, but apparently that's another aspirin substitute, and that also contains an amide. They act to reduce fever and pain, but have little anti-inflammatory effects. All right. Other amides are contained in barbiturates, which is a type of drug that are kind of known as like sedatives or hypnotics. So they can be sedatives in small doses or even like sleep inducers in large doses. They tend to be habit forming. So a lot of sleep medication, Luminol, Nembutal are all contain amides. Those are barbiturates. And again, they're basically make you go to sleep, right? Sedatives. Okay, in terms of hydrolysis of amides, right? So we talked about how you can form amides from carboxylic acids. Now we can go the other way, which is to break an amide up. And so this can happen either as a base catalyzed reaction. So using NaOH and heat, we can cleave, and notice where it does cut. It cuts between the nitrogen and the carbonyl carbon, right? So we're breaking it back up into our carboxylic acid. In the presence of a base, it would actually form the carboxylate ion and our amine, right? And so this is a hydrolysis reaction that occurs under basic conditions, right? So since it's a base, I don't get my carboxylic acid, right? Because this guy wouldn't exist under basic conditions. I get the amine, or I'm sorry, the carboxylate ion. Likewise, because I'm under basic conditions, I will get an amine and not an ammonium salt, which we'll see under acidic conditions. Okay. Again, importantly, it's that nitrogen carbonyl carbon bond that's cleaved. We could do the same thing under acidic conditions, and we would get a very, very similar sort of products in the sense that we're still cleaving the bond in our amide bond in the same place. But because we're under acidic conditions, we would actually get the carboxylic acid on the other end. So it still has that hydrogen bound. And because, again, we're in acidic conditions, we get an ammonium salt, right? Because amines act as bases, they'll accept protons. So if we have this under acidic conditions, it's going to accept a proton and thus have that positive charge, being an ammonium salt. Okay, so you can have either base catalyzed and acid or acid catalyzed hydrolysis. It's always cleaving at that same place between the carbonyl carbon and the nitrogen. The difference is under acidic conditions, you get a carboxylic acid and an ammonium salt. Under basic conditions, you get a carboxylate ion and an amine. Okay, yeah, so this is just another example of the base catalyzed hydrolysis. Again, cleaving at that carbonyl carbon nitrogen bond. Because it's under basic conditions, I'll get a carboxylate salt where my oxygen will be deprotonated and have a negative charge. And because it's under basic conditions, there's no protons for this amine to accept. So it's just an amine. All right, so draw the condensed structural formula and write the IUPAC names for the product of the hydrolysis of N-methylpentanamide with NaOH. Okay, so first of all, N-methylpentanamide. So let's just pick this apart. My parent chain is a pentanamide. So that's 1, 2, 3, 4, 5 carbons. And then, so that takes care of these 5 carbons. And then I have this N-methyl. So that means there's a methyl group off of my nitrogen, and I have to fill in what would be missing, which would be the hydrogen. And so this is going to be a base catalyzed reaction, adding NaOH. So base catalyzed hydrolysis reaction. Okay, so again, importantly, where do I cleave? Right, how am I going to chop my amine? Right there. So I will have a 5 carbon, either carboxylic acid or carboxylate. We'll sort of think that out in 1 second here. Those 5 carbons, so hopefully you can tell on either side of the reaction where those 5 carbons came from. And because this is basic conditions, I'm going to have the carboxylate salt. Okay. The other portion, the nitrogen that's bound to its single carbon. And because we're under basic conditions, whoops, that's not what I meant to do. Because we're under basic conditions, so there's that carbon, it's going to remain the amine. And so this would be meth and amine. Okay. Cool, so checking our work looks good. Again, importantly, cleaving at that nitrogen carbonyl carbon bond. Because it's under basic conditions, we will get a carboxylate salt and an amine. All right, so now let's draw the products of this reaction where we have our amide in the presence of hydrochloric acid. So now this is the acid catalyzed hydrolysis. Again, where am I going to cleave? I'm going to break this molecule apart there. So that means I'll have 1, 2, 3 carbons in my carboxylic acid. And it will indeed be the acid form because we're under acidic conditions. And I will have my nitrogen that's still bound to its 1 carbon. Okay. But now, because we're under acidic conditions, it should be the ammonium ion. So how many hydrogens will I need to fill in to create the proper ammonium ion? Right, so I'm going to need 3 hydrogens and a positive charge on that nitrogen, right? Because the ammonium ions have 4 bonds. Cool.